we get into the plasias. Plasia just means a development or formation. It's also from the same root as plastic, which is something that can be molded or changed. Okay, so we have different types of plasias. The first one we're going to talk about, it's pretty straightforward. Hyperplasia. This is mainly just remembering the terms. Hyperplasia is an increase in cell number. Okay. Metaplasia is a change from one cell type to another. Okay, so in this you can see here is normal, and then we see atrophy where they got smaller. Okay, and then we see hypertrophy where they got larger, and then hyperplasia is where they increased in number. So you can see that there are more of them here. And then metaplasia, you can see that they went from sort of this cuboidal to maybe this squamous epithelial tissue in this particular picture. So that's metaplasia. Dysplasia, dys meaning like it's in dysfunction, deranged cell growth. So that means that they may have started out looking like this normal one up here, but now you can sort of see that these no longer look like that, not even a little bit. These are kind of completely, they're becoming an unknown cell type. They're not normal at all. So that's dysplasia. Now remember, for all of these things, that you had to do some kind of a DNA manipulation. You had to do some kind of a gene expression. You had to turn off some genes, turn on some genes, whatever. But you had to mess around with how your DNA is expressing genes to make proteins, because you've changed so much. And that's where these things can a lot of times be considered precancerous, because a lot of times they will progress from hyperplasia to metaplasia to dysplasia. Not all the time. Sometimes this is perfectly normal, and they just go back. Okay, they go back to where they were, and they go back to the normal cell once the stressor is removed. But sometimes they don't. Okay, and we'll talk more about that at a later time. Okay, so first of all, hyperplasia, the organ may become larger, but it's due to more cells, not larger cells, as you would see in hypertrophy. It's almost exclusively found in cells that are capable of mitotic cell division. Well, this just makes sense. If the cell can't divide, well, then you're not going to have hyperplasia. You just can't. If a cell is like nerve cells or muscle cells that don't divide, well, you're not going to have hyperplasia in those cells. However, if you do have cells that can divide, then hyperplasia is possible. Okay, so it's not in muscle cells and neurons. You don't see hyperplasia there. Examples: regeneration of a liver after partial hepatectomy. So you remove part of the liver, and then that will trigger growth signals that will come in. Growth factors will trigger the proliferation of the existing cells that are there. Okay, breast enlargement during pregnancy. So that's a hormonal trigger that says that you have to start producing and enhancing the glandular cells that were there to begin with, but now this hormonal signal says that you need to increase the number of those because they are about to be used. Calluses are another example of hyperplasia, normal hyperplasia, and when you remove the stressor, if you're digging a pond in your backyard, whatever you happen to be doing, you may develop calluses, but then once that stressor has gone away, then your hands will get all smooth again and the calluses will leave. Okay, so hormones may affect the endometrium or the prostate gland. Endometrium estrogens may cause that, prostate gland androgens may cause that, and you may get some hyperplasia going on there. And so that's and you can see in this picture, there's a normal endometrium, and then there's endometrial hyperplasia, where this endometrial lining has expanded. You've gotten more of the cells that make up the endometrium.